So my talk today is focused on that floriculture section of the research and extension I do. Okay, um, I work with a lot of growers in Indiana and also in Midwest, and I consider myself fortunate because the conversations I have with growers, the discussions I have with growers, really inform me the real world problems that they're facing. And so what we do is we bring those questions to the table and ask ourselves, do we have any ready-made solution already out there from published research? And if it is not there, we do our own research. We come up with high quality research and we provide those research, the information to our growers. Not only just providing information, we work with our growers, we train them in technologies, sort of being doing things in more responsible way. Um, Today's topic, smart sensors for floriculture, kind of came out of these type of conversations. Um, Sarah mentioned I used to work for Monsanto company. So basically, Monsanto company is heavily involved in, as you know, crops like corn, soybean, cotton, field crops. But then what I learned while working at Monsanto is already in outdoor production, field crops, this type of precision ag, digital ag, or smart agriculture, this is already there. It is actually commercial in outdoor agriculture. This technology is becoming easily available to farmers. But then if you look at this floriculture industry, very little research has been done. There are companies that are coming out, but most of our growers are not sure if this technology is going to work. And even if they wanted to get this technology, some of these technologies are very expensive, complicated. So when I started at Purdue University, I asked this question myself, how can we make such a useful technology easily affordable to our growers and make it such that it is so simple to use, but most importantly, as a floriculture grower, some of you are floriculture growers, you have to make 100 decisions a day, and let us help you with a few decisions just to make this your job easy. So that's kind of like the motivation for this research. Okay, um, so here are the topics. Here are the topics that I'm going to go over today. Um, Now, in terms of demonstration, perhaps we'll see if we can get to that in the end. If not, I can always refer you to a link on my website where you can actually see this demonstration. But what I want to do is I wanted to talk about why plant monitoring is important in floriculture. And then I want to talk about what are the issues with plant monitoring, and then dive into the main topic, what are smart sensors, and what can you measure with these smart sensors? How did we develop this technology? And last, and probably most important, is how can we make this technology available to floriculture growers? So those are the main topics, okay? And the demonstration piece, probably I might simply refer you to a link. I'll probably show you that link on my website towards the end. It's a very easy piece of uh, using these sensors. So it's a fairly simple demonstration also. Okay, so that said, let's start the conversation. So if you notice my slides, you will see that most of my slides have a lot of pictures. I make sure that my slides are usually simple. Here and there, you may find some graphs, some scientific data, but they're also very simple graphs. But whenever there is a graph, I will slowly explain it, okay? But most of the stuff is very simple, good pictures. And then I usually have a sort of like a concluding statement at the top of every slide that kind of emphasizes what I'm going to talk about in each slide, okay? So I wanted to start by saying that floriculture is intense production. In other words, the investment in terms of dollar amount per unit area is very high. The amount of resources, lighting, temperature, nutrients, automation, robotics, um, and also not, it's very important to keep in mind the number of plants per unit area is also going to be high. So the investment on seed, plants, containers, everything is quite high. So high resource oriented production. So that's important. And you can see the different types of floriculture crops. Um, if you're not familiar with this particular industry, but I will mention in the United States, the top ones are bedding plants. And then followed by bedding plants, we have container plants like poinsettias, chrysanthemums, and Easter lilies. Um, Industry is kind of growing more in warmer states like California, Florida, Michigan. And what we notice there is also some new crops coming out. For example, succulents are becoming quite popular, especially among millennials and Z generation. It's very easy to grow them. So I think that's probably one the reason they like it. Also, these are tough and hardy, and youngsters really like to see that, you know, them or see an image of those plants, tough image of the plants, and kind of that's probably why they probably like it. But the industry is approximately about $5 billion wholesale value in the United States, dominated by bedding plants like petunias, marigolds, um, pansies, impatiens. These are the top 4 or 5 bedding plant species. Okay, hanging baskets have become quite popular, like probably even in Canada nowadays. And so they are you'll see hanging baskets everywhere. A lot of crop species are grown in hanging baskets nowadays, but some of the popular varieties like petunias are still the most popular ones. Okay. Uh, Chrysanthemums are moms, you can see in this particular picture towards the right. It's not grown inside a greenhouse. Chrysanthemums are grown outside. They usually start in July and have these plants ready around this time. Okay, but several thousands and thousands of pots you can see in this picture. This is one of the greenhouses in Indiana, um, showing up chrysanthemums. Okay, chrysanthemum pots. So that's a little bit introduction about floriculture, but keep in mind the key point I want to mention, it's a high resource investment business. And some of you probably know that. Now, if you are running a business and where you're investing a lot per unit area, this is where monitoring becomes critical. And I'm showing you some examples here, and I want to emphasize that point again using these examples. And these are real pictures, some of growers who sent us, and some of the pictures are also from my own research. If you don't monitor, for example, let's say here is a plug producer. On the left side, I'm showing you a tray of seedlings, pretty much failed germination in this case. And this particular produces almost 1 or 2 million seedlings during that January, February time. And he wants to make these seedlings ready for March spring planting. Now, a little bit of carelessness, little bit of lack of monitoring can result in a situation like this. That's a total loss. And you can see almost 60–70% loss in germination. That means he's not going to be able to meet the requirements of his clients, mostly landscapers, right? So monitoring becomes so critical. The amount of plant seedlings he's producing, it really makes it challenging, but it is critical. In the center picture, you can see chrysanthemums. This is one grower who recently contacted me. He was thinking everything is fine. His electrical conductivity, which is a term we use to measure the overall nutrient status or fertilizer status in a container, it measures the total amount of fertilizer salts dissolved in the nutrient solution inside a pot. And that is a very easy measurement. And he was actually showing me that these EC numbers are actually looking great. The pH was great, but here you go. Still, you see the parts showing up yellowing, some micronutrient deficiency here likely, but parts showing up yellowing. And he has this showing up in 10–15% of the parts. And I've shown you a picture. Usually growers in Indiana and Midwestern states have about 200–300,000 of these chrysanthemum parts. 10% to 20% of parts showing this type of damage is critical. So that continuous monitoring probably could have helped him detect those nutrient disorders probably earlier. Okay, and also poor growth. I mean, if you're not providing water or light or nutrients or temperature, you can start seeing plant growth significantly affected. Poinsettia containers at the top, and zinnia plants at the bottom. And we just did a A study where we asked this question, what happens if you reduce 30% of water, 30% of nutrients, or 30% cooler temperatures? What happens to crop? And we were seeing that there's a significant effect. Environment plays a big role. And if you don't monitor and if you don't maintain optimum environment, above ground environment, it can also produce really bad quality plants. That means poor quality plants means lack of profits, less profits. You may not even be able to sell them. So these are the kind of things: intense farming, high resource investment, and really challenging to monitor because of thousands and thousands of plants, but yet so important to remain in business. Okay. So just want to emphasize, set the stage with all these slides. Okay. All right, so then what's the problem with monitoring? So if you have to monitor, let's monitor. Well, it's not that straightforward. Keep that number in mind: thousands and thousands of plants, seedlings or container plants, or plants in 4-inch containers or liners, whatever that. There are so many of them. Some of the monitoring techniques, for example, I use the word EC, electrical conductivity. It's a very important measurement. And one of the techniques we use is called pour-through method. It's basically, some of you probably follow this, and you can see steps here. You water the plants, and then afterwards you place some saucers below these parts, add a little bit of RO water or fertilizer water, and then you collect the leachate. You collect the leachate, so that additional water you add at the top of the container pushes the water that is already inside the container. You don't want to add too much of water. You just want to add a little bit of water. And then you collect the leachate, and you take that leachate into a saucer, and you measure the electrical conductivity using a EC/pH meter. So yeah, you can monitor nutrient status of the plants, but then keep in mind, this process there are several steps already here, right? And then in order to complete this pour-through method, it takes almost, per part, and on average, it takes, 4 to 5 minutes to get some good data after thoroughly irrigating and waiting for an hour, and you start this process. So imagine and multiply that with the number of plants you have. How many plants should you sample? I asked the same question to one of our growers, and a chrysanthemum grower basically told me that I don't have enough labor, so we just probably monitor 10 to 15 parts in a 3 or 4 acre plot where I have 200,000 chrysanthemums. Is that right number? Such a tiny sampling, so you may not know anything what's going on with your plants. So that's one thing to keep in mind. So the techniques are labor intensive, time consuming. The second issue is, a lot of our growers, and it's probably the case in many countries, they kind of take the path of visual assessments. Well, there's nothing wrong with this visual assessments. It's just that, those people who are making this visual assessment should be really trained. I mean, look at this picture. There are 2 poinsettia containers. Yes, there is some yellowing on the leaves. So kind of it shows that it's probably some nutrient disorder, but what nutrient? Is it nitrogen? Is it sulfur? Is it magnesium? I mean, I can see some intravenoal chlorosis here. I was starting to think nitrogen, but then top leaves are showing yellow. Usually bottom leaves show yellow in color. So unless you have that type of experience, you cannot actually pinpoint and identify nutrient disorders with visual assessments. The other problem with visual assessments is you can only assess when the symptom becomes visible. It's a visual assessment. So by the time you make an assessment, it may be too late. You probably cannot save that plant or save the group of plants. In floriculture, if there are some leaves that are not looking good, that's not a high quality or a good quality plant. So you cannot probably sell that. So visual assessments also have some inherent issues. In addition to that, the person making these assessments should be very well trained. With pandemic, I'm pretty sure it's the same case even in Canada. The trained workforce, it's a huge challenge here for us. And so it's not that easy to find those people who are really trained. Even before pandemic, so the workforce was always an issue in floriculture industry. So finding those talented people who can make accurate measurements is not that straightforward. Then we have our standard laboratory assessments, right? We can take a sample, send it to a laboratory, get it analyzed in a laboratory by specialists using some analytical procedures. Here is an example of plant analysis report that we actually use even in our research. We send samples and they send different percentages of different elements, and they also send us whether the element is higher or lower or optimum levels inside the plants. Great stuff. Again, this is the standard. But then, like I described for the other assessment methods, the laboratory measurements also have some issues. It takes about a week at least. You got to collect the samples. Some of the laboratories actually ask you to dry the samples, kind of grind them, send them. And if you don't grind or dry, yes, they will do it for you, but it costs more, $5 more per sample if you don't grind it. But then just to get a report like this, what you're seeing here, 13 or 14 elements inside the plants, it takes about a week and it costs about $20 per sample. Now, how many samples can you send it? And if you're a large scale producers, how many samples should you take? Those are all the challenges. And in addition to that, there is some additional work, right? For sample collection, sample preparation, grinding, shipping, all those things. In floriculture, especially with that high intense production, you want to know the answers today, not a week later. It may be too late, right? So I'm trying to, as you see, I'm trying to build my case here, step by step towards that smart sensors, okay? Just bear with me for a few more slides and we'll get to that. Smart sensors, okay? So the next thing is there are equipment, great stuff. Immediately you can get some reasonable information about what's happening to your plants. You have sensors that measure nitrogen status. You have sensors that measures the temperature of a leaf. So when the leaf is warmer or colder, you can adjust the temperature in the greenhouse to make sure that the plant is, again, what plant experience is what matters. I know there are researchers who monitor environment. That's great, but then end of the day, what is in the plant is what matters, right? So you can actually measure the plant temperature and adjust the greenhouse settings based on the plant temperature. You can also measure, chlorophyll and a surrogate of nitrogen status of plants using SPAD measurements, like the one that I'm showing you in the middle. And if you have money, you can also buy really expensive hyperspectral cameras and get a lot more information about their growth, other things like color progression, things like that. But then look at the numbers at the bottom, those dollars numbers, $15,000, $2,500, $1,000. Many of our floriculture growers are small to medium scale, 3,000 square feet greenhouses. Some of them, just doing business during spring and fall. If I go and showcase this type of technology, show them that this is great, probably they may like to hear from me, but this probably not going to be, really attractive to them because of the price, right? Always, in any business, most of the growers are looking for an investment. The investment is something that can give them a return within 3 to 5 years. That's the key here. So can I get a return if I invest $15,000 on a hyperspectral camera? It probably will take 10 years to get the return on that, right? So there are sensors that can directly measure plant statuses like, temperature, nitrogen status, growth, things like that, but they are very expensive. So that brings us to smart sensing. So what are smart sensors? So far I've mentioned about a few things, couple of things. Monitoring is critical, but monitoring is challenging. The available techniques either are time consuming, expensive, or it can take a lot of labor, cumbersome procedures. So how can we solve this problem of plant monitoring? So we've been thinking about it and we came up with 2 types of sensing techniques. One is solely based off your smartphones. Take a picture and receive the information that you're looking for on your phone instantaneously. Isn't that attractive? Very nice, right? We'll talk more about that. The other one, you see at the bottom, this is more like a IoT, Internet of Things type sensor. This is more like a stationary one. You can place this sensor on top of plants. You can see a picture of poinsettia trace images of poinsettia plants captured by this small, IoT sensor. And these sensors cost about $150 to $200. They're about 4 inches by 3 inches in size. This can be placed on top of the plants at stationary positions. They are programmed to take images and send those images to a central computer. You can program it to send every day, every week, every second, whatever you want. And they'll send the programs to a computer, and in the computer, all these images of the plants will be processed. And same information like you get on the smartphone, you can also receive that type of information on your computers. So we've been building these two types of sensors. So what are the advantages? First of all, they are based off of plant images, okay? We capture image, we don't even touch the plants. So these are kind of remote in their nature. So we take images of plants and we extract the information that is of economic importance to our floriculture growers from those images. And I'll show you what that type of information is very quickly. But there are other few things I wanted to mention about these smart sensors. They're low cost. I mentioned to you that these IoT sensors are probably $150 to $200. The smartphone-based sensor is even cheaper. I mean, it's just a licensing fee, and I'll talk about that towards the end. So they're really low cost, nothing like $1,000, $2,000, $15,000 range. They're a few $100 range. So way cheaper. They're easy to use. Like, everybody knows how to take a picture on your phones, right? It's that easy. Everybody knows how to use an app on your phone. That's how easy it is. Even these IoT sensors, you don't have to do anything. Just put it on top of the plants and they'll take care of everything. As long as you have internet in your facility, they'll do the job for you, nonstop, day and night. They're easily adaptable. That's the key thing here. They're attractive. They're less cost, the low cost. And that's why we think it's highly adaptable. And last but not the least, let's say you have a situation where you don't have a lot of trained people in your facility, but then you don't have time to train them. You can use these type of smart sensors to actually make people get trained. I mean, these smart sensors can tell them, they can take a picture and right on their phones, they'll see the information about the plants. Right there, they are learning. For, let's say they're walking and they see a plant that is not looking so good. They take a picture of the plant and the smart sensor tells them that there could be something wrong in terms of growth or something else going on with the plants. Then they can relate what they have seen in the facility to what really is happening as shown by that smartphone sensor. So that's the training piece. You can train untrained workforce. You can rely on these sensors so they can be trained. They can use this. Everybody has got a phone. All you need to do is get them this license for these apps, and they can, as long as they work, they can use this. Junior growers, senior workers, anybody in your facility can use this facility, this technology, okay? So this should give you some introduction about what smart sensing is. The word smart is a catchy word, like a smartphone, right? And it's actually not, these pictures are actually being used to sense some of the useful stuff that we are interested in. That's the reason why these are called smart sensors. All right, so what can we measure? The first and foremost thing is plant growth monitoring. Now, don't worry about these pictures or the graph, we'll get to them, but I wanted to give you a little bit more information what plant growth monitoring is. Now, let's say you have a group of bedding plants, okay, marigold plants, or pansies, or petunias, or impatiens, and you want to see how these plants are growing. Are they tracking the right growth rate that is desirable to you? You want to finish the crop maybe in 4 weeks, maybe in 6 weeks. Is that growth rate of these plants tracking the path that will lead them to that harvesting stage within 4 to 6 weeks that you originally intended? This is very important because you have already some contracts with landscapers, and you want to make sure that the plant is finished the time when they need it. And so growth tracking is so important. Now, there are many ways you can do. You can cut the plant, you can weigh the plant, or you can take the plant and research what we use, something called leaf area meters. These are big scanners. We run these plant leaves under these leaf area meters. We kind of know how much area is there, and from that area we can see, the plant is growing normally. If you do this 1 or 2 years, you can actually develop your own growth curves. I use an analogy like, when you take your kids to a doctor, what the doctor does is they get their height and weight of these kids, right? And then what they do after that, they get the height and weights and they plot their weight and height on some standard charts. And then they tell you, yeah, the kid is growing okay, yeah, 50 percentile height, 100 percentile weight, whatever that is. So they're tracking their growth using some standard charts. Exactly like that you can do for your plants. 1 or 2 years down the line, you can have your own growth curves for your own facility using this technology, and you can track the plant growth. It is so important. When does my tomatoes get to harvest? When does my, I kind of digressed here because I also work in the area of vegetables, but let's stay with floriculture crops. When is my crop going to get finished? Is it 4 weeks or is it 6 weeks? That extra 2 weeks is critical. So how does this happen? Now let's get to this picture. So here is a picture of plants. I just took this picture the other day in my backyard. Basically, you can see this black and cloud parts. But then I want to point you to a red square here. You see that red square? That's a standard square. It's just a piece of paper. That's all, school paper, as simple as that. And I cut it precisely, and that square is approximately 16 centimeters square. I know that that is the area of that red square, okay? And then now I put that piece of paper next to the plants, and imagine doing the same thing next to the group of parts, okay? And then you place your camera, or you have this IoT sensor I've showed you on top of the plants. You take a picture. As soon as you take this picture, what happens is we have already built in this software, and that software knows what is a plant and what is a background in that picture. You see this picture that's called segmented plant? Now you don't see that bag or that red square or even the substrate, the media or anything. All you see are those green leaves. The software can identify what is a leaf, what is a plant, and what is not a plant. Now it does with almost 95%, 98% accuracy. If you have a media which has got a lot of white color, then could be some issues. But in general, if you use a peat-based media or even coir media, it's not at all an issue usually. But then afterwards, what happens is it separates, throws away that background, only counts the plant-based pixels in the image. And then it also does at the background, what is the standard I'm showing you here is a standard. The same thing, like it identified the plant, it also identifies the standard. And after identifying the standard, it knows the standard is certain area, and now it uses that information to estimate the area of the plant. Now, why are we measuring the area of the plant? The area of the plant is directly related to the size of the plant. The area of the plant, top-down, two-dimensional area of the plant, is also in many, many species directly related to the weight of the plant. Weight and size. By size, I mean, the volume, the expansive growth, things like that. These are the things we care about, right? When we talk about growth, we want to know the weight, we want to know the size of the plants. So when you take this picture, you instantaneously get that canopy area, which is the top-down area of the plant. In this particular case, something like 2934 centimeters square, approximately 2900 centimeters square. That is the size of the plant today. You come back 2 days later, you take a picture, you get a bigger size. 2 days later, you take a picture, you get a bigger size. But then you keep doing this. You keep doing this. Now what you're doing is you are tracking the growth of your plants. Okay, you do this many, many locations in your greenhouse for many, many species. You kind of see how the growth is changing in different locations of the greenhouse. When you see in some locations it's faster and in some locations slower, you kind of know something else is going on in regions where the plants are not growing faster. At least now you know there is a problem. Even before you visually see, you can identify the problem. We actually have data that shows that we can see growth-related issues 3 to 4 days before human beings can actually visually identify them. That's simply because our eyes are not that sensitive. We cannot see something that is like 3 or 4% different. Our eyes are only sensitive to something that is like 10, 15% different. So we're not that really good at visualizing, bottom line. But this image-based technique will do that for you. Now, I also want to show you this graph. Now, it's been there, and I've taken a lot of time explaining this, but when we do the research, we're not just doing something and saying that this is the canopy area you should trust. We always calibrate, validate it against a standard. Now, research standard is this leaf area meter. That's what we use in my lab and many other labs. What we have done is we've taken these images, we estimated the area, And then we took the same plant, separated the leaves, ran all those leaves through that leaf area, standard leaf area meter. And we also get the actual leaf area. That's what I'm calling measured leaf area. See here, the measure. And this is the estimated by the smart sensor. And you can see the relationship. It's a nearly one-to-one relationship. And this R square basically means it is pretty good fit, okay? So we can now estimate the actual area just by taking a picture. By the way, that actual leaf area meter is $3,500. We get that information with the phone, okay? That's the power of this technology, all right? This is just one example. Growth monitoring, tracking growth. I'm going to go a little faster now that I've introduced you what this is. I'm going to go a little faster now. See here, this is another popular thing that we always get asked. People want to know about nitrogen status in plants. You know, if the way you normally they get the nitrogen content in plants is they take a sample, send the sample to the laboratory, wait for a week, pay for $20 per sample, and they get a measurement of plant nitrogen status. But we have developed software, same smartphone based, take a picture. That software, you know, looks at how much of the light from the red band is reflecting, from the green band is reflecting, and the blue. And these are all the kind of things that are associated with plant physiology. Plants absorb red light and blue light in photosynthesis, and we know that. And if the plants are not absorbing either red or blue light, mostly if they're not absorbing a lot of red light, we know that there is some problem because they're supposed to absorb red light in photosynthesis and grow better. So we are looking at all those things. All this stuff happens behind the screens. You don't need to you even see all of these things. End of the day, you take a picture. You can see here is a picture of salvia plant. I just took it with my phone, quickly process things, and it throws something called an index, a nitrogen index, N index, okay? I want to show you, here is again some validated calibration data. Here is our nitrogen index from our smartphone, right? And here is a SPAD measurement. SPAD is, I'm going to go a few slides back, and I wanted to show you the picture of that instrument. I should have put it here on the slide, but let me go back and I'll show you that quickly. So here it is. This is a SPAD meter. It is a chlorophyll meter that can be used to measure plant nitrogen status indirectly, and it costs $2,500. Keep that number in mind. So that is that SPAD measurement. So on the same plant, we measured SPAD measurements. We also took pictures and measured our nitrogen index, almost with 70% accuracy. And this data is coming from 4 different species. And if I plot these responses for individual species, you know, you'll see R square as 0.8, 0.85. But we want to have a sort of like a universal relationship. Does it work across species? And it does. And you can see for many species that smartphone measurement is as good as a SPAD measurement. We didn't leave it there. We went further and we looked at how is our smartphone measurement related to the standard laboratory measurement. Remember I showed you a laboratory measurement analysis report that you get for each sample. We did the same thing. We sent the same sample, sent to the laboratory, waited for a week, and they sent us back some numbers. And then we calibrated and we looked at across all species, 4 species. There is a nice linear relationship. So basically, what I'm showing you on this slide is with those simple smartphone measurements, that N index value, nitrogen index value that we estimated from that image I've shown you in this picture. Remember this N index coming out from just a picture? That is related to data collected by an expensive equipment and even nicely related to standard laboratory measurements. So that's the power. So now you can use this measurement with almost 70 to 80% accuracy. You can collect pictures of the plants as many times you want and get information about nitrogen status of the plants. When the nitrogen status falls below a threshold for the species, you can start your fertigation. And that's kind of optimizing the fertilizer application or optimizing fertilizer rates. Now you are basing it on the plant nitrogen status, not basing on EC, the one that I described. You remember the pour through method? It takes about 4 or 5 minutes per plant. This one, click of a button, within few seconds, you will see the nitrogen status of a group of plants. You walk away to another section, measure 100 pictures, 1,000 pictures a day, it's up to you. But that's the power. The more data, the more analysis, the more assessments, the better estimate it is, right? And here is the power right in your arm, in your hands, inside your smartphones. That's the power of this type of technology. Few more things. If you're a plug producer, one of the main things you're worried about is germination counts. I've shown you a picture where seedlings have, germination was poor, right? What do you need to do? You need to know how many seedlings have germinated so I can refill those trays and still, sell my produce. If you are a seed producer, like a seed company, you need to know the seed quality. Germination percentage is one of the primary characteristics you measure. But then it goes to the point again, to the quantity of measurements you get. If you get 3 or 4 trays, yeah, just count them, come up with germination percentage. But let's say you have 15,000 trays and you wanted to get an average germination percentage. If you're a plug producer, you want to make sure that you have enough number of seedlings and you have 15 to 20,000 trays. How do you get a good estimate of germination percentage? Just take a picture like I've shown you, and you can see the same picture, marigold seedlings, and the software counts them and throws you germination percentage. This happens in 1 second. So you can take your phone, walk, as you're walking, stop second, take a picture, go another area, take a picture, maybe take 100 pictures and kind of get a very good average estimate of germination percentage. In 1 hour, you can get a lot of data. But if you're counting and calculating with the calculator, you may probably do 10, 15 trays. After that, you get tired, bored, right? That's another useful measurement. Now, if you're a bulk flower producer, and I'm giving an example of anthurium flowers here. One thing you want to know, gerberas, anthuriums, roses, anything that is red, yellow color, what you can do is you need to know how many flowers you can harvest in a given day. And yes, you can stand in front of a section of flowers, you can mark the area, you can take a picture like I've done here, and the software can count and tell you how many flowers are there in that particular area. Now it is able to identify what is a flower versus what is a leaf. It throws away leaves, it only picks those flowers, and it counts the flowers and throws the number of flowers. And this can be a highly economically useful for bulk flower cut flower producers. They want to know how many flowers they can harvest, right? And this is one way to do that. You can count them, of course, but you don't have to walk inside or do anything. You can stand several feet away, mark an area, take a picture, and then move on to the next section, take another picture. Every time you do that, you kind of get number of flowers in that unit area, and you can do that over and over and over again, okay? Another important measurement, plant growth regulator application. Now, many of you apply plant growth regulators to control the height of the plants, right? To make sure that plants are grown in containers are compact, good quality, and you can ship them without any issues. In India, and I've seen actually some of the growers who actually track the height of their plants using a ruler. So they walk into the, let's say, I'm sorry, in this particular case, I'm using poinset example, and they want to make sure that they apply plant growth regulator right time. They don't want to apply too early or too late, right? You want to apply plant growth regulators when plants are growing actively. And how do you know if they're growing actively? They measure the height. They track the height from week to week. And if there is a steep increase in height, they know that, okay, then now is the time to hit them with the plant growth regulators, right? But then the way they do that even today is take a ruler and walk inside, measure the height, record the number on a piece of paper, come back, draw it, and maybe plot an Excel graph and see what's going on. But you can take a picture of the plant or groups of plants from the side using your phone, and you can see here the same plant, just took a picture, and within a second, it gives you the height and width of the plant, height and width. And not only just that, it takes that information, plots onto a graph, and you can see how that height is changing, okay? Over time. So everything automated very, very fast. I got only a few more slides. I won't take more than 2 or 3 minutes here, but I wanted to show you things that are very important. We develop technology based on real experimentation. It's not just coming out of some software guy coming up with a program and throwing onto a smartphone. We validate using high quality equipment. Like you can see here, a multispectral image station is like $25,000. We use these type of equipment to actually measure everything, kind of figure out what is the right thing to measure, and then take that piece onto the smartphone. So before this technology gets on the smartphone, a lot of pilot work is done, okay? Experimentation, replicated trials. Here is a trial of poinsettia plants or bedding plants grown at different levels of nutrients in a tightly controlled sub-irrigation system. We wanted to create the range of values in plants, and then we collect the plants, we do statistical analysis carefully. All those things are taken care of before this technology actually gets to the commercial stage, okay? I just want to mention that. Even with software processing, you don't see all those things. A lot happens actually from the point you take an image to the point where that image can be processed. You know, step by step, a lot of things you can see here, you know, as you go through, images are now how the background is taken care of and removed before that image of seedlings can be used for further processing. So a lot happens in the background. But we don't want our growers, our clients, users to get confused with all of that. You can just look at this technology and ready-made information is right on your phones. So how are we going to make this technology available? Now, the few things I want you to know, if you're really interested in technology, remember this word, Aptus, A-P-T-U-S. There will be an app, and we're going to advertise this broadly. Perhaps I'll also send this information to Sarah and others. Aptus stands for Analyzing Plant Traits Using Smartphones, an acronym, okay? Aptus is a Latin word for apt, appropriate, right? This is the right word. So Aptus is the name of the app that we're going to release in spring of next year. We're at a point where we have a lot of data. We're just putting together everything into an app form, so you can just download the app. There's going to be some license fee. I'm going to talk about that briefly. But then you can use the app. And once you click on the app, the first thing you'll see is the app will ask you what type of species you're interested in, poinsettia, hydrangeas, zinnias. Then you select your species, then it'll ask you what type of trait you're interested in. I'm interested in nitrogen, you select nitrogen. I'm interested in germination counts, flower number, you can select that. I'm interested in growth, you can select area. Once you select that, it provides you some instructions on how you place a camera. Some of you probably have deposited your checks in a bank, isn't there? Nowadays, which I don't even go to my bank. I've just put the check and then I place my phone on top of the check, and you know the phone tells me move the phone camera up or down, and then eventually takes a picture, something like that. It'll guide you how to take a picture. It's very important because we want to make sure that you take pictures consistently from same height, same angle, so the data is consistent. Those are the kind of instructions that show up. And afterwards, you just take a picture. After you take a picture, what happens is for each grower will have their own username, login information, so that your data is safe. I don't see all of the data, nobody. So it's all highly protected. But then what happens is once you log in and you take a picture, the data goes from your phone to a server maintained by Purdue University. That's where all the software, our intellectual property is. And the server processes these images, sends the information back to your phones. That's how we want to do that, okay? Randomly, we may take a few pictures randomly without knowing where the pictures are coming from, just to see how the whole thing is working. And is there anything we need to optimize further? Are there any issues with these technologies? Other than that, we even don't know. But this is what we are thinking of, licensing this technology. There will be some license fee. We're doing it. We want to do it on an annual basis. But again, keep in mind, it's going to be minimal. Remember, a simple sample you send to lab might cost $20. With this technology, you do it 1,000 times, million times. It's, in a year, it's all yours, right? But then the reason why we want to charge you a little bit is that money will be used to do more research, maybe hire a graduate student, conduct more research, develop more traits, add more species, things like that, okay? I'll stop there, and I want to just acknowledge a couple of my graduate students, Ranjita and Cheng, were very instrumental. Kirby is one of the IT guys who was initially involved in this project. But then also big thanks to the 3 foundation industry donors, sponsors in the United States, Fred Glowickman Foundation, American Floral Endowment, and Horticulture Research Institute. All 3 came together. There's a lot of money involved in this project, and we needed support from all of them. So I'll stop there. We have 8 minutes left. I don't know whether I went too long, but I'll be available if there are any questions to answer. Should I leave this? I'll leave the deck in case there are questions. We'll come back.
Okay, yeah, I stopped sharing for now, but we can always go back to sharing and looking at slides if
Okay.
particular questions come up. But yeah, thank you so much for that talk, Dr. Namali. I didn't realize like how easy and accessible that could be for growers, that kind of technology. So that was really fascinating for me. And Siobhan, I'm sure you've been watching the chat and come up with some questions of your own, I'm sure, for Dr. Namali.
Yeah, I think we've got a few questions. So how this is going to work, we'll walk through the questions in the Q&A section. So if you have a question for Dr. Namali, just make sure you type it in there and we'll get to it as we go. If you're having difficulty, you can message me through the chat, but try and keep those questions in the Q&A section if you can. So Dr. Namali, if you're ready, we could start with the first question.
Sure.
So we have a grower who wants to know, will the app work for perennial varieties?
We are conducting research on nursery crops also. We have worked a lot on hydrangeas, and the app works perfectly for hydrangeas. Now, the growth tracking measurements, that is kind of universal. That can be used on any species. Yes, certainly you can't take a picture of the tree and kind of get a good estimate of it. But, things that you can grow inside greenhouses, that'll totally work fine for that. When it gets to analysis like nitrogen status in plants, things like that, yes, it can be used for any species. However, if we haven't conducted research, all you're seeing is a raw nitrogen index number. And then you have to figure out what that number means. Now, there are a lot of people who work with me. They want to do something similar on conifers, evergreens, tree species that I don't conduct research on. But what they want to do is they just want to use this app, take measurements, and they want to conduct research of their own in their own facility, figure out the meaningful value of an index that they need to be targeting to grow healthy plants. And once they figure that out, they just want to maintain that particular index value for that species. So they want to do it on their own, which is totally fine. But what we do is we have a set of species that I already worked on. I worked on a lot of bedding plants. You've seen a lot of data on poinsettias, chrysanthemums. I'm working on lettuce, many herbaceous species, hydrangeas, hostas. But then again, in research, we are limited by resources and time. So over time, we'll add more species. But whatever we do, what we do is we develop these nice algorithms. So when you take this nitrogen index measurement, it will give you actual percentage of nitrogen in plants rather than an index value. So we've already calibrated that. So you can actually use that information directly. But yes, you can. It's a long answer to your question. Definitely, if you are one of those who wants to, do a little bit more, probe more, and do your own research, absolutely, you can play with the app. It's only going to give you a number, and so you can come up with your own way. If you haven't done the research on that particular species, you can do it yourself.
Okay, that sounds good. It sounds like lots of growers could customize this for their operation, which is great. The next question, you talked a little bit about using the app to identify nitrogen deficiency. Mm-hmm. Do you have any plans to include other nutrient deficiencies?
Yeah. We do have plans. And I'll mention a little bit more about this nitrogen deficiency piece. The way the nitrogen deficiency is being measured is it's measuring indirectly a pigment called chlorophyll in plants. Okay. And chlorophyll, not all of the nitrogen in the plants stays in chlorophyll, but there is a significant amount of nitrogen in chlorophyll. And that is what we are trying to capture. If there is nitrogen deficiency in plant, it should also be seen in chlorophyll. Okay. For example, the lot of nitrogen in plant resides in an enzyme called rubisco. But we're not measuring anything with the rubisco. We're looking at that particular pigment. Okay. So there are some challenges if you are working with pigments. If you're a researcher, you have to look at both sides of the coin. So what are the challenges? Well, chlorophyll levels in plants are also affected by some other elements than nitrogen. For example, if you look at the chlorophyll molecule, biochemistry of chlorophyll, right in the middle of the chlorophyll, magnesium is there. Right? That's the central molecule. What it means is chlorophyll contains magnesium. And if you have magnesium deficiency, you will see something with chlorophyll, and then that might throw as a deficiency number, but that's probably not coming from nitrogen, but it's probably coming from something else. So what we are doing is we have done a lot of research already, what we call model development. We look at the effect of individual elements on this particular nitrogen index. And what our research shows is this particular index, what we have developed, is affected mostly by nitrogen. Okay, 75% of the variability in that measurement is coming from nitrogen, but there are also minor things coming from other elements like magnesium, iron, sulfur, zinc. By the way, all these elements are related to chlorophyll biosynthesis in plants. So we know that there are other elements that are impacting this measurement. So we just need to figure out how we can separate those measurements from nitrogen to get an estimate of micronutrient deficiency in plants. So that research is ongoing. We are actually conducting right now some research to fine tune. What we do is we nail down, zoom in on certain wavelengths that these plants are reflecting, what we call as crop signature. It's like, in simple terms, we're talking to plants, okay? Not in English, not in their language. Their language is a signal, reflectance that comes out of the light that reflects back is their language. And we are picking that, we are trying to interpret what that means. We just need to fine tune, understand the grammar better to figure out what's actually going on with the plants. I think we should be able to, our next element of interest is phosphorus. And it's because there's a lot of pollution happening in our natural ecosystems, like Lake Erie has a huge phosphorus issue, and a lot of people are asking me to do that. So there is ongoing research on identifying other elements as well. It's just a matter of time. I will say this much before I know there's a long answer, but there are only a few people out there who are very specific about elements, but in general, in production, nitrogen is a dominant fertilizer, and all of the rest of the things that we apply to plants are based off of nitrogen concentration, right? You take a fertilizer 20, 10, 20, and the recommendation is 150 ppm nitrogen. But then if you're applying 150 parts per million nitrogen using that fertilizer, you're also applying everything else along with it. It's just not nitrogen, right? These are all complex fertilizers. So from the production side, as long as you're managing nitrogen, you're almost always, unless there is a very specific issue, you're almost always good. But we are trying to also come up with some distinguished signatures for phosphorus, nitrogen, and perhaps some of those micros that showed up significant in our original analysis.
Sounds good. Sounds like there's some work to be done there.
Yeah.
So that's great. And I guess the purchase of the app would help to fund that research. So that's
absolutely. It is true.
Yeah. A couple more questions. So a lot of our growers are interested in sensor technology to monitor how their crop develops year to year. So different weather conditions or different pressures or timing can affect how the crop develops over time, say this year versus last year versus 5 years ago. And they're trying to use that data to fill a certain number of orders as they go ahead. So would this technology allow growers to track how their crop is progressing year to year? Can you compare that information in the data?
Exactly, and that's one of the biggest advantages, right? I mentioned about the growth tracking piece, but basically what happens is it will take a year or two for a grower to develop their own standard curves. Okay, let's say this year has been great, and you're really satisfied with plant growth rate, but then you've also been collecting your data this year with the smartphone, okay? That becomes your standard growth curve until the next time you see something better than this. But what you can do is you can use that growth curve year after year, season after season, and start comparing the growth curves from other times with this particular growth curve. We are trying to actually do that right on the app. So what's going to happen is when you receive the data also sits on some form of an Excel file or some form of a sort of like a graphing device. That's what we're coming up with. And so you can see the numbers, but then the number is going to be plotted onto the graph. So you can see how the change is happening. But then we will also add in a standard that something like a customizable thing that you can pick up 2019 growth curve as your standard. So it plots that as another curve on the same graph. So you can actually start seeing how the plant growth from the current season is matching with the best one that you have produced. So that's the nice thing about this technology. And those are all easy things has been done. Taking a data and plotting on a graph is fairly simple. The real complicated thing is the science piece, and then making sure that you're collecting the right type of images. I will say this, and the reason why I kind of mentioned this when I was showing you, how the app works. There's going to be a set of instructions to users on how to position the camera and take a picture, and that's critical because, especially if you're doing this growth tracking, you want to make sure that the distance between the top of the plant and the camera kind of remains similar. Of course, we are normalizing everything with standards, but for accuracy's sake, we wanted to make sure that that's always followed and the angle is also properly kept. So that's one reason why we provide the instruction. But also in those instructions, we'll also indicate what we are seeing is that it's not with growth tracking, but for nitrogen assessment measurements, it may be really good to take the images either in the morning or in the evening times and avoid those afternoons where the fluctuations in light intensity are really high, cloud passage or high light levels, things like that. They may throw some confounding effects. And what we're seeing best is when you take these measurements in the evening or in the morning, when the fluctuations are relatively less in terms of natural sunlight intensity, you get the best type of data. So those are the kind of things that we're building into the app itself. Those are the instructions that we tell our users. But a little bit of that, not too complicated. Height and light intensity are the two major factors we are working on. The rest of the stuff is fairly straightforward.
It's great if that's noted in the app. I know a lot of our growers, myself too, we've got lots of things on our mind. So if those instructions are nice and clear, it makes the app easy to use. And like you said, like a training tool, right? For new staff. Yeah. Or transitional staff in your busy season, things like that. People who might not know all of those little tidbits as we go along, right? So that's great. One other question, you talked a little bit about how you could use the app for plant growth regulator application timing. And I wondered if you're, again, making that growth curve for your crop, can the app predict for you when you would be able to apply an application if you give that information to the app? So if you say, I normally apply 3 applications 2 weeks apart for a crop that looks like this, can you give that data to the app and the app is able to predict when those applications need to be done? Or is that something that you'd be collecting data for a couple of years until you get to that point?
Yeah, it can be done, but we haven't included that feature in it for one reason. It's more a personal choice. Some growers want to wait a few more days until they get to a certain height before they want to apply PGRs, while others are much more data-driven. So it's a personal choice. Adding that type of information is fairly simple. Basically, you're saying that when height of points areas is 25 centimeters, hit them with this particular growth regulator. But that is just a recommendation. So we want to leave that option to users because our floriculture growers are experienced growers. They've grown this crop. What we are really trying to do is making their job or work less, making sure that their measurements are accurate, easily collected. But then afterwards, they can make their own choice. I mean, when the height is this much from last year. So this again something that they can learn from year to year. Last year, I've applied PGR when the software was showing the height of 25 centimeters. That was not good. So I'm going to try different next year. So that's something that will probably evolve with time. So we wanted to leave that option to our clients, customers, as opposed to telling them to do everything. Yeah.
Sounds good. Do we have anybody else who would like to ask a question? I don't see any typing. Thank you very much for talking to us today, Dr. Namaly.
My pleasure.
We really appreciate you sharing your information with our growers. So just to clarify how to get a hold of the app is called Aptus, A-P-T-U-S.
Yeah.
And you expect that it would be released in spring 2022?
Correct. That's
Right.
the plan. And we'll
And-
use a lot of venues commonly seen by growers, floriculture growers, national and international venues. Yeah.
And would it be released for both Android and iPhone?
That's our goal. Yeah.
Perfect. Yeah.
It's actually
Excellent.
easier to do on an Android than iOS, but we are trying to do it for both.
Excellent. Very good.
Excellent.
Uh,
Yeah, well, thank you again for coming and talking to everyone and for answering all our questions. And I'm sure we'll be hearing more from you in the future because it sounds like you have a lot of exciting research to share with Ontario.
Absolutely. Thanks for the opportunity. I really enjoyed talking to you and answering some of those questions. It's a very important question.